That's my job. Dropping the ball. Okay, there you go. Down a little bit.
All right, great. Thank you again, Travis, for that introduction, and good job to Tim Lawrence. Kind of a hard act to follow, so I'm a little bit glad that the bee porn video didn't work, because I really would be hard pressed to follow an act like that. So we're going to take a different but related subject matter on to task right now, and this is a project that a graduate student of mine in my program has been doing, and he's not here today, so I'll be presenting on his behalf. And what we're looking at for this particular project is if foliar nutrients, specifically calcium and boron, can help improve fruit set in high bush blueberry. So as I mentioned earlier in my previous talk, blueberry is capable of setting 100% of its flowers and having fruit develop from that, but 80% is considered a full crop. And here in Western Washington, we can have poor conditions that lead to only 50% or 60% of our flowers actually setting fruit. And this represents a pretty significant and substantial loss of potential yield. There's also some cultivars, Draper being among them and the most notable one, that have been exhibiting premature fruit drop. And so reduced fruit set and yield could be due to pollination, which I addressed previously, and it could also be due to plant nutrition, specifically boron and calcium. So why the interest in boron? Well, just a little bit of background information is provided here. So boron is an essential micronutrient. Its essentiality was established in 1923. So we've known that for some time. It's a metalloid chemical element, and one of its primary roles is in cell structure. It also is involved in a whole host of other things like nucleic acid DNA synthesis, protein synthesis, formation of sugars. I could go on, but really what we're interested in from the perspective of fruit set is its role in reproductive processes, especially pollen health and pollen function. And the available forms that we have for foliar nutrient applications is boric acid and tetrahydroxyborate. So we may be prone here in Western Washington to boron deficiencies. And most agricultural soils, we range from 1 to 467 milligrams per kilogram of soil in total boron concentration. But here we tend to see lower amounts than that. I know in some of my own blueberry planting at the Mount Vernon Research Station, we have about 0.47 milligrams per kilogram of soil of boron. And so we're right at the cusp of being deficient there, according to recommendations coming out of Oregon State University. Some symptoms of deficiency that we might see if it's deficient in our plants are tip dieback, aborted leaf and bud development, and then we might see increased winter injury. But there might be a more hidden deficiency that we might not be able to see in our plants because of its role in reproductive health and pollen. So inadequate boron might reduce the health of our pollen, its viability, its ability to function, and its ability to function in helping us fertilize our seeds and setting our fruit. So I wanted to just give you an idea of what sufficiency levels look like in high bush blueberries. So we have on the left are the various concentrations from blueberry leaf tissue samples collected between mid-July and mid-April or mid-August, and the various status that those leaves can have based on the concentration. If deficient, the current recommendation is to apply a foliar spray before bloom or after harvest, but before leaf drop. But I also want to take this opportunity to warn you that if you are using applications of boron to help increase fruit set or correct any deficiencies, to be careful, because there's a fine line with boron between deficiency and toxicity. So there are several factors that influence the availability of boron. From the soil perspective, the first is organic matter. So organic matter in our soils is an important reservoir for boron, and temperature affects decomposition and subsequent release of boron. So we have temperature extremes, either too low or too hot, that affects decomposition and subsequent release and availability of boron to the plant. Soil texture is also known to have a big influence on boron availability. We see decreased amounts of boron being available to plants under coarser texture soil conditions, and then more boron being available to plants under more higher clay content soils. pH affects the nutrient availability of most all nutrients, and for boron, they are more available to plants at a slightly acidic to neutral pH, so pH of 5 to 7.5. And then weather is another factor that can influence boron availability. So dry and cold weather can reduce root activity in our plants and their ability to take up boron. Cold temperatures can also influence the mobility of boron in the plant, so getting it from the roots to a site where it is needed, where the demand is. And then high rainfall, which we have here in Western Washington, can also cause leaching of boron from our soils. So there's a lot of research, well, there's been some research done with other Vaccinium crops and the role that boron can have in improving fruit set and yields. So in cranberry, there has been documented evidence that if we apply foliar applications of boron, we see increased fruit set and yield in those plants. And I'm talking about cranberry because it's very closely related to blueberry. It's in the same genus. Now in lowbush blueberry, even more closely related to highbush blueberry, there's also been some studies down on the East Coast where applications, both fall and spring applications of boron, can again increase fruit set and subsequent yields. And then there's another interesting study where it documents with lowbush blueberry that if we're applying boron, we do see increased pollen germination and seed number. And this points to the role that boron has in allowing pollen to the pollen tube to grow. So pollen germinates once it lands on the female part of a plant, and it needs to grow down that long style and then fertilize those ovules so that they can develop into seeds. And the boron seems to be most important in allowing that pollen tube to grow. There have been some studies with highbush blueberry, and really the results have been mixed, and those studies have been done in Michigan, they've been done in Missouri, they've been done even in Germany, but we haven't seen anything here in the Pacific Northwest. Now I want to shift to calcium and talk a little bit about why I'm interested in calcium. So calcium is an essential macronutrient. It's needed in larger amounts than boron, which is a micronutrient. And it's really known for its critical role in cell structure. But it also has several other important functions in the plant, helping promote membrane structure. And membrane structure is really important for the functioning of a cell and overall health of a plant. And I'll talk a little bit more about membranes in my discussion on cold hardiness. But it has other physiological roles, like involved in nutrient uptake, enzymatic relationships, just helping the cells in a plant function and helping the plant perform. And the form that is used by plants is calcium 2+. So deficiency is actually rarely seen in highbush blueberry for our conditions, and most tissue samples range within the recommended levels of 0.4 to 0.8%. But distribution of calcium can be very variable within a highbush blueberry plant. It's also very variable across varieties. Draper is a great example. We can see high levels of calcium in the leaves, but very low levels of calcium in the fruit relative to other highbush blueberry varieties. And calcium has also had some demonstrated role in as a dip in increasing fruit firmness. We really haven't seen that in highbush blueberry if it's applied at a field through field applications. So now factors affecting calcium availability. So pH is one that affects availability of calcium in your soils, and low pH can lead to low calcium availability to plants. And whereas higher pHs can lead to greater availability of calcium to plants. So if you're a grower and you're having calcium deficiency and you suspect it might be due to the pH, the recommendation is to collect leaf tissue samples during that recommended time frame, mid-July to mid-August, and look at manganese in your plant tissues. And if the manganese level is 450 parts per million or greater, it's very indicative that the low calcium problems that you're seeing might be due to a pH issue. Then I'm going to breeze through some of the rest of these here. So cation exchange capacity, that's your soil's ability to attract, retain, and exchange nutrients like calcium. That influences calcium availability. Weather, so high rainfall can lead to calcium leaching, and that's why a lot of our soils in Western Washington are actually naturally pretty acidic. A lot of our basic cations like calcium are leached away through the rainfall. And then there's plant factors, and I mentioned some of these already, but high vigor is associated with lower calcium levels in the leaves. Reduced transpiration limits calcium, and this is due to the fact that calcium moves in the xylem or the pipes in the plant that move water to various organs in the plant. And if we have low transpiration, like in our fruit, then calcium is not being moved to that tissue because it's being pulled through the pipes in that plant. And then distribution, as I mentioned, varies across cultivars. So now I want to build a little bit more of the justification in terms of why we're studying calcium. I talked about the functions that it has in the plant and some of the conditions that influence availability of these nutrients. Well, there's reports, as I mentioned, that boron and calcium applications can increase fruit set and yield, and we've seen that in cranberry, we've seen that in low bush blueberry. It's also applied and seen in other horticultural crops, like almond being among them. Both nutrients are found in pre-bloom sprays, and I've had a couple of growers and distributors ask me about the role that these nutrients have in affecting fruit set and yield in blueberry, and I frankly can say I don't know because we have not done the studies to justify whether or not these are available and absorbed into the plant. Sufficiency levels, as I mentioned, might vary between leaf tissue and reproductive tissue like pollen, and that's a very emerging field. So just because our leaf tissue concentration of boron is sufficient doesn't necessarily mean that we have enough for our pollen to get fruit set, particularly if we have conditions that limit fruit set already, which we do to have here in Western Washington. So the objectives of this project in terms of looking at foliar applications of these nutrients is, in the short term, to determine if foliar applied boron and calcium can increase fruit set and yield for Washington blueberry, and we're specifically looking at here in Western Washington. Now, I only have 1 year's worth of data to present on, but this project and other related projects will contribute to the long-term goal of trying to generate information in terms of justifying the use of these products for growers, and also to help achieve optimal ways of applying these nutrients and optimal timing. So there's 2 experiments that I'm going to discuss. The first experiment was done with established Duke plants at the Mount Vernon Research and Extension Center. We collected soil samples just to get a baseline understanding of what the nutrient levels were in our soils, and then to see what influence that it had on plant growth. We also wanted to make sure that our plants were starting off with a boron deficiency before we started these treatments. And so we collected the samples, both leaf and tissue, and verified boron was not deficient in the leaves, and we proceeded forward with the experiment in fall of 2014, and we continued application in the spring of 2014. And the design, it's a randomized complete block design. We have 3 plants per plot, and we had each treatment replicated 3 times. And what we applied is on this slide here. We had a control, which was no foliar nutrient applications. Then we had a spring applied low boron treatment, and we had a spring applied high boron concentration treatment, fall applied low boron, and fall applied high boron. And the treatments were applied according to the product manufacturer's recommendations. We didn't exceed the levels that was being advised according to the manufacturer. And the specific product that we used was Nui Boron 10%. This is a boric acid boron. And this was a product that we chose based on conversations with growers in this area, that this was one that they'd be interested in seeing some trials on. But I'm really sorry to say that we don't really have very good data for this project for this year. So we observed a lot of blueberry shock virus in our plants for this year. So we were able to collect some data, but I want to mention this right out from the beginning so that some of the results, I would interpret it cautiously because we had high levels of shock. So with that, though, we did expand the project, and that's going to be discussed more in experiment 2. And we included 4 sites for this second project that I'll be talking about. 2 in Whatcom with the variety Draper, and 2 in Skagit County with the variety or cultivar Bluecrop, and that was on an organic site. So for the second experiment, we wanted to look more at calcium and boron, and then calcium and boron in combination. And the products that we used are here shown on this slide. Our treatments were applied to 5 plant plots this time, and they were replicated 4 times. And we looked at a high boron treatment using Solubor at 6 pounds to the acre, a low boron using Solubor at 3 pounds to the acre, a high calcium using Phytocal at 4 quarts to the acre, a low calcium treatment at 2 quarts to the acre, and then we used the combination treatment, high boron plus calcium at a high concentration. This was a Phytoset product at 4 quarts to the acre, and then a low boron plus calcium product, again, a phytoset at 2 quarts to the acre. And lastly, we had an untreated control. If you're wondering why we use some of these products, well, we use Solubor, Phytocal, and Phytosets because we had some organic grower cooperators, and we wanted to test these products on their field situation. And in addition, we're working with Dr. Bernadine Strict in Oregon, at Oregon State University. He was also using this product. So by utilizing the same products, we get some similar information. We can see the efficacy of these products and these nutrients in blueberries. It allows us to study these nutrients more. And the applications were made at the early pink bud stage, and they were reapplied every 7 to 10 days for a total of 6 applications as per the manufacturer's recommendations. Now, some of the data that we collected soil, foliar, and fruit nutrient, tissue, and fruit nutrient concentrations. We also looked at fruit set and estimated yield, berry size, fruit firmness. We looked at fruit firmness specifically because there's some belief that calcium applications can increase firmness of blueberries. Seed number, premature fruit drop, and then pollen viability as well as pollen health. So the lower picture on the slide, those are extracted seeds from our treatments, and we were able to count the number of seeds as well as discern high quality seeds from low quality, like the darker ones, indicating that that seed wasn't properly fertilized or something in the developmental process didn't allow it to complete its development and produce viable seed. So the first year data for this project, as I mentioned, we want to interpret this cautiously because the shock in the first experiment at the research extension planning. But with that said, we didn't see any statistical significant differences in fruit set, estimated yield, or fruit firmness across all of the treatments. As I mentioned, our soil test ranged, well, said 0.47, 0.32 to 0.44 milligrams per kilogram of soil and boron, and we're at a deficiency level if we're below 5 milligrams of boron per kilogram. And then our tissue concentrations ranged from 42 to 63, so we were in that sufficiency range. One thing we did note, it wasn't statistically significant, but plants treated in the fall had a higher boron concentration than those treated in the spring. But we really need to collect more data and repeat this study next year in order to monitor this. So these are some of the data that I'm presenting here. The figures are a little bit reduced, but the message here is we're looking at fruit set on the top left and estimated yield at the lower right. We did not see any significant, statistically significant differences. And again, I surmise that what we are seeing in terms of the low yield, again, it's not statistically significant for the spring low boron applications is due to that being heavily infested with blueberry shock virus. So for experiment 2, and I also want to say, researchers might be very cautious. I want to say we want to interpret these data cautiously too, because we only have 1 year's worth of data, and 2015 was a very unusual year. We had low rainfall and we had very unusual high temperatures. And I think these factors influence the data very much. That being said, we did not see any statistical differences in fruit set, estimated yield, or firmness. We also didn't see any premature fruit drop in Draper. And that was corroborated by several growers that they saw very little premature fruit drop. And this was both in Western Washington and British Columbia. And for those of you that aren't familiar, Draper has exhibited premature fruit drop. So at the greenberry stage, right before it transitions into developing color, fruit can drop, and some growers have reported losses of up to 40% due to this premature fruit drop. And Mr. Eric Gerbrandt at University of Saskatchewan has been doing studies, and it seems like the problem is due to calcium deficiency. And he has seen in some of his trials, application of calcium chloride can mitigate and reduce Draper drop. We didn't see drop, though, in any of our treatments. We also didn't see it in our control, which had no applications made. So that leads me to believe that Draper might be due, and well, calcium is related to the premature fruit drop issue, but there might be environmental factors as well, such as solar radiation and temperature, solar radiation being among them, and how that affects the carbon balance of the plant, and that might manifest into more or less fruit drop. We did see a statistically insignificant trend for increased fruit set with the boron and calcium treatment. We did see little uptake of calcium in the leaves and the fruit, and that's also corroborated by some studies that Dr. Strick has been doing at Oregon State University. But we did see also that cultivars seem to respond differently to the various treatment applications. So I'm going to go through some of these data. I just kind of gave you the highlights. Now I'm going to show you the data. So here we're looking at fruit set from the second experiment. And we have percent fruit set on the y-axis, and then we have our various treatments. And I coded this just to make it a little bit easier in terms of what the control, what the low boron, high boron, low calcium, and etc. were. And then if you're interested in varieties, the orange and the gray are Bluecrop, and the yellow and the blue are Draper. And so here what we see is, again, really statistically insignificant data here. However, what I do want to point out is that our fruit set is, for the most part, above 80%. We had very unusually high fruit set. Again, I believe this is due to the weather that we had, high temperatures, good conditions for fruit set and fruit development. And these slides are reduced, but here what we're looking at is fruit and foliar concentrations of boron and Bluecrop and Draper treated with our various boron and calcium treatments. So here we have the fruit boron concentration on the left graph and the leaf concentration and the graph on the right. And the blue and the red bars here are Bluecrop, and the green and the blue bars represent Draper. And here what we see is, it looks numerically like there's differences, but the data were pretty variable, and statistically we didn't see any differences in leaf boron concentration. What we did see in the fruit, it was statistically insignificant, but there was an interesting trend that we did see up to 60% more boron in the high boron treatments. And also Bluecrop seemed to be more sensitive to the boron applications. And we can see this in that the fruit and the leaves tend to have more boron than the Draper in both leaf and fruit. So now we're looking at fruit and foliar concentrations of calcium in Bluecrop and Draper treated with foliar applications of boron and calcium. Again, same layout and organization here. So on the left, we have fruit concentration of calcium, and on the right, we have a figure with leaf concentration of calcium. And here what we saw, statistically, no significant differences. There's numeric differences, but when we ran the stats, there were no differences. And we did see, actually, this was pretty promising. It wasn't statistically significant, but we saw a modest increase in fruit calcium in our treatments, up to 22% in our high calcium treatments. And there was also, we observed, similar to Dr. Strick, which she has observed in previous studies, that Draper had more calcium in the leaves. It also had more calcium in the fruit. So conclusions from this first year, again, first year of the study, we had unusual weather conditions, so it's hard to draw really strong conclusions. I firmly believe we need to repeat the study another year, and I'm really torn. I hopefully wish that the weather would be a bit more marginal so we can see how these products perform under more normal conditions that we have here in Western Washington, the cool, wet conditions where fruit set is limited. And again, we believe that the results were highly influenced by the weather that we had for this year. So with that, I want to acknowledge a few people. So first, I want to acknowledge the grower cooperators that we worked with for these projects. Sean Watkinson, he is the technician for my program. Lee Kausis, he is a apple tree physiologist in Wenatchee, and he works quite a bit with calcium in apple. So we work closely with him and try to see about nutritional needs of calcium in blueberry. The Watcom Farmers Co-op, and then funding for this project came from the Washington Blueberry Commission. And so with that, I've left some time for questions. Do we have any? Yes.
Will help with
Sure. So the question was, I heard it. I can repeat it though for the benefit of the audience. So the question was, do I believe that any of these products will help prevent berry shattering? And so can you define berry shattering for me first too, just so I understand what you're saying here? Okay, so pre-harvest drop of berries. So there has been some preliminary data that has shown that calcium in various forms, calcium chloride being among them, can limit premature drop of fruit. Boron, I don't, understanding the physiology and how it functions in the plant, I do not believe that that would influence shattering or premature drought. Yes.
Oh, preliminarily, from your data, would you say that a foliar application of boron or calcium is stored in the plant and made available the following spring?
Sure. So that's an excellent question. And that's one reason why we have spring and fall applications in the first treatment. Preliminarily with the data, so the physiologist in me says I really don't believe that the boron would be stored in the plant. However, some of the preliminary data, we did see higher amounts of fruit set and boron in the plants with the fall applications. So at this point, I would say it's plausible, but I can't definitively say at this moment. Mm-hmm. Mm-hmm.
Okay. Thank you,
All right.
Dr. DeVetter.
Thank you.
And if anyone filled out their surveys, please submit it at the table at the end if you can. Thank you.